Alrighty, welcome to the small intestine, section 24-6. Let's start with the anatomy of the small intestine. So the small intestine follows just after the stomach, and it comes in three portions. First the duodenum, then the jejunum, and then the ileum. There's some fun mnemonics online if you're struggling organizing which comes first. The duodenum is relatively C-shaped, and it's also the shortest out of the three. Then you have the jejunum. The jejunum is going to be the longest out of the three, and that's where most of the absorption is taking place. And then you have the ileum, which is about as long as the duodenum. Between the ileum and the large intestine, the ileum will connect to the cecum, which is the first part of the small intestine. And there's a junction in between the two called the ileocecal valve. And this is a valve that's going to, a muscular valve that will allow passage of pre-feces from the small intestine into almost feces into the large intestine. So the histology of the small intestine, we've got lots of transverse circular folds, which we've talked about for the digestive system, functions to increase the amount of absorption area or surface area that we have in order to increase the amount of absorption we can get from those products. And it also allows for expansion, so intake of greater volume of a bolus or chyme or at this part of the digestive system, feces. Each fold will have intestinal villi, and each of those intestinal villi will have microvilli on top of those villi. So that's what I have drawn over here. So if we take the tube and we zoom into a part of the tube, then we'd see villi, and we'd see simple columnar epithelium. And if we were to zoom in closer to that, into those villi, we could see little microvilli on top of that, and those microvilli will serve to increase the amount of absorption that we can possibly get. So here we go, these are microvilli. And the microvilli will have something called a brush border surrounding it, that I'll make in green here. And we'll talk about the brush border's importance here in a second, but let's just label it for now. And if we can imagine where that brush border is in relation to the small intestine, it's a very thin layer. It's kind of, I shouldn't say a very thin layer, but it's very closely associated with the intimate layer of the small intestine. It'll, I don't even want to give it a length, but it'll only touch the microvilli of the small intestine. It won't go anywhere past really into the lumen, and that'll make more sense whenever we talk about its functionality. Okay, each villus will have its own lamina propria and its own vascular network in the submucosa. So we can draw, let me go in another shade of red. So each one will get its own artery, each one will get its own vein, or venule. I mean, these are really small. And each one is also going to get something called a lacteal. You may remember lacteals whenever we were talking about the lymphatic system. Lacteals are super important for transporting fats from the diet, or from the small intestine into the lymphatic system. And we want to separate the lipids that we get from the that we're absorbing. We don't want to quite put it into the bloodstream quite yet because we need to package it into something called a chylomicron. And then when we package it into a chylomicron, then we can put it into the blood vasculature. Otherwise, we would just have a bunch of lipids floating around all the time, having no targeted area of where they can go, and they would have, and then they would clog up arteries much easier. Okay, we also have this artery and vein for each villus, and that's really important because that's how we're going to get the nutrients directly into the bloodstream. We need to have this really intimate relationship and really close blood supply in order for us to be most efficient in actually getting all of those nutrients absorbed and put them directly into the bloodstream. So here's some nice pictures of what's going on. We have the mucosa, submucosa, muscular layer, and serosa layer, just like before, except here we need to differentiate what's special about the small intestine versus other portions of the digestive tract. So we mentioned those lacteals. We also need to mention that we have specialized cells called Paneth cells, and these are going to be in the the intestinal glands. Paneth cells are going to serve as immune cells here. They'll be sampling the environment and making sure that everything's all right. If it's not all right, then it'll call in the immune system to come and help. We'll also have a bunch of stem cells in this area. Stem cells are really important because this is our primary area of where we get absorption, and if we don't have any absorption of the nutrients, then we die. So we need to make sure that this epithelium is well taken care of, and if any of the cells are dying, we need to be able to rapidly replenish them, so we're going to have some stem cells in this area. All right. We'll also have some goblet cells sandwiched in between the columnar epithelial cells. These will be important in making mucus, so we still have some mucus produced in the small intestine. It's just to help get things moving. At the base of each villi, we'll have those intestinal crypts. Like I said, we'll have stem cells there, and we'll have Paneth cells. Paneth cells are named after someone. I'm not sure who it is. Then we'll also have enteroendocrine cells. And enteroendocrine cells are really important because they're the ones that are going to help coordinate the release of gastrin, CCK, and secretin. So once enteroendocrine cells are activated, gastrin in the G cells of the stomach will G cells of the stomach will release gastrin. CCK will be released from the eye cells, and will tell the gallbladder to start contractions and pushing its chyme into the duodenum, and also telling the small intestine to start contractions as well. And then secretin will tell the pancreas to release bicarbonate in order to help deacidify the chyme as it enters from the pylorus into the duodenum. We'll also have lots of submucosal glands here, which are making extra mucus, and we'll talk about where those are. So functions of the duodenum, we're receiving chyme from the stomach and we're neutralizing acid. This is basically a big goblet that's accepting everything from the stomach, from the pancreas, and from the biliary tree, so from the gallbladder and the liver. And it's a relatively short area, but we're having a little bit of absorption in this area. Then we'll have the jejunum, and the jejunum is going to have a lot more circular folds and a lot more villi, and that means that this is the primary area of absorption. Then we have the ileum, and the ileum doesn't have as many circular folds, just like the duodenum, but it does have Peyer's patches, and if you remember, Peyer's patches come from the immune chapter, and these are regulatory cells that will sample the environment of the lumen of the small intestine and recruit the immune system if things are haywire. So one easy way to differentiate what's going on in the slides for each of these, I'll draw down here. So the duodenum is going to have few villi versus the jejunum is going to have very, very, very long villi. And then the ileum, you'll see Peyer's patches. The duodenum, you'll also see some mucus. Oh goodness, oh goodness. Mucus glands. So whenever you're looking at a slide and you've identified that it's probably the small intestine, you know it's not the large intestine because the large intestine is going to be a lot more flat in the cells. There's not going to be a lot of divots. So once you've identified that it's the small intestine, in order to differentiate between duodenum, jejunum, and ileum, I would start with first, is it the jejunum? And you can identify the jejunum really easily by they'll have really, 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 really long villi. If it's not that, then do you see Peyer's patches? You know, these are these big, like, purple goblet looking, or not purple goblet, but purple big nodule looking cells. And if it's not one of those two, then you're probably looking at the duodenum. Okay, so let's talk about the physiology of the small intestine, starting with chemical digestion, then we'll talk about mechanical, and then we'll talk about how we regulate all of this. So starting with chemical digestion, we need to talk about the brush border enzymes. So here we drew the brush border, and let me draw in pink where our brush border enzymes would be. So these are going to be only existing in the brush border, and they are simply waiting there for the inactive proteases from the pancreas to arrive so that they can turn on those inactive proteases help digest some of the proteins at the brush border, and then it'll be easier for it to absorb at the in the small intestine. So we call those brush border enzymes enteropeptidases, which makes sense. ACE for enzyme, pepti, we're referring to breaking down peptides, and entero, we're talking about the small intestine. So these enteropeptidases take trypsinogen and convert it into its active form, which is trypsin. And then trypsin can go on and activate procarboxypeptidase and turn it into carboxypeptidase, and whatever other inactive proteases we have coming from the pancreas. But trypsin is the active form in order for us to start breaking down some of those proteins. We'll also secrete bicarb here to help deacidify the environment. We'll release enterocrinin from the enteroendocrine cells. This is just helping promote all of the hormones do what they need to do. We also have parasympathetic innervation here. This is going to be initiated by the cephalic phase, or during the cephalic phase, where we're releasing mucus. All right, mechanical digestion. Again, we're just doing peristalsis in this area, not really a lot of segmentation. We're primarily trying to get stuff out of the duodenum into the jejunum, keep it in the jejunum for a bit, and then send it on its way. We have the reflexes that are active that also involve the small intestine. So anything that distends the small intestine will initiate shorter reflexes to help contract it again. Anything that involves but if there's something in the small intestine, it will promote the small intestine to continue contractions, continue peristalsis, to move things down the GI tract. So regulation, we have both parasympathetics and sympathetics here. Sympathetics are going to stimulate peristalsis. Parasympathetics are going to not stimulate peristalsis as much. And of course, we have those reflexes, as we mentioned last time.